Today we will discuss bacterial transduction. Transduction is the process by which a virus transfers genetic material from one bacterium to another. These viruses are called bacteriophages, and bacteriophages are able to infect bacterial cells and use them as hosts to make their progeny. Right? After multiplication, these viruses assemble and occasionally remove a portion of host cell DNA, that is bacterial DNA. When one of these bacteriophages infect a new host cell, this piece of bacterial DNA may be incorporated into the genome of new bacterium, right? So transduction is of two types, generalized and specialized. Bacteriophages are obligate intracellular parasites. Obligate parasite means an organism that cannot complete its life cycle without exploiting a suitable host, right? Obligate parasite. When phages propagate, they can sometimes encapsulate host bacterial DNA to form transducing particles. These transducing particles are ostensibly like mature phage particles, and they inject only bacterial DNA into the new host cell instead of viral gene, right? The DNA can then recombine into the chromosomes or replicate as a plasmid into the new host cell. So this process of transferring bacterial DNA from one bacterium to another is called genetic transduction, right? Since their discovery in the 1950s, all fog-mediated gene transfer was thought to occur by two well-described mechanisms, generalized and specialized. However, with the recent discovery of lateral transduction, there are now three modes of fog transduction: generalized transduction, specialized, and lateral transduction. So, because transduction is a fog-mediated gene transfer process, so first we will discuss what is the structure of this bacteriophage and how it multiplies. Bacteriophage have a very simple structure. You can see here head and tail, and these are the tail fibers. So genetic material of bacteriophage is contained in this prism-shaped head surrounded by protein coat, right? This is connected then to elongated sheath by a neck or a collar region. This sheath forms a hollow tube through which the viral DNA or RNA is injected into the host bacterium or host cell, right? At the bottom of this sheath is the base plate to which tail fibers are there that facilitate attachment to the host cell, right? Now, in order to multiply, phage must first enter the host cell. So first step is phage attachment to the host bacterial cell to specific receptor on the cell surface, right? Then create a hole, right? Then rigid tube is propelled out of the sheath, puncturing a hole in the bacterial cell membrane, then through which phage inject their genetic material. This red in color you can see here. Then inside, viruses take over the host cellular machinery for their own replication, right? If surrounding conditions are unfavorable, they undergo a process called lytic cycle, lysis, right? But if the conditions are favorable, under unfavorable condition, lytic cycle is there, but under favorable conditions, virus may enter a dormant state. And this type of the cycle is called lysogenic cycle. Now first, what is lytic cycle? Here, infecting fog ultimately kill the host cell to produce uh, their own progeny, right? So there are 4 steps of lytic cycle. First is fog attachment. Second is bacterial cell entry. Third, fog replication. And the fourth one is birth of new fog, right? Immediately following the injection into the host cell, fog genome synthesize early proteins. Then what is the function of these early proteins? They break down the host DNA. You can see here in this diagram, right? So the fog then uses the host cell to synthesize the remaining uh, components, remaining proteins required to build new fog particles, right? Then head and sheaths are assembled separately. New genetic material then packed into the head and new daughter fog particles are constructed. During this process, host cell gradually become weakened by fog enzymes, and eventually you can see here lysis in the lysis phase. Eventually, bacterial cell burst, releasing on average 100 to 200 new fog progeny, right, into the surrounding environment. So this is lytic cycle. Now lysogenic cycle. Lysogenic cycle, also known as temperate or non-virulent infection, it doesn't kill the host cell, right? Instead, using it as a refuge where it exists in a dormant state, right? Following the injection, first virus injects its genome DNA into the host cell. This integrates itself into the host genome. This red in color, right? So with the help of Phag encoded integrase enzyme. So then prophag genome replicated passively along with the host genome. As the host cell divides for as long as it remains there only and does not form the proteins required to produce progeny. As the phag genome is generally means comparatively small, bacterial hosts are normally relatively unharmed by this process, right? So first we will discuss generalized transduction. So generalized transduction first discovered in Salmonella phag P22. So it is the process, generalized transduction is a process by which phags can package any bacterial DNA, chromosomal or plasmid, and transfer it to another bacterium. Transducing particle of this mode of transduction form when bacterial host DNA is packaged into phag heads instead of viral DNA. So here all the fragments of donor DNA from any region of chromosome have a chance to enter into transducing particles, means bacteriophage. Here in generalized transduction, first bacteriophages infect donor cell. When virus enters into bacterial cell, it takes over host cell and synthesizes virus components such as viral genome, viral enzymes, capsid, head tail, etc. These viral enzymes hydrolyze the host cell DNA into small fragments. These small fragments. So during the assembly of virus component to form new viruses, sometimes any of the fragments of donor DNA get incorporated into virus capsid. Such abnormal bacteriophages when infect a new cell, it can transfer this donor DNA into new bacterium. Then what will happen? Because this donor DNA is not viral DNA, so it doesn't replicate inside the recipient bacterium, but undergoes recombination, homologous recombination with recipient bacterial cell. right? So new type of the cells are formed called recombinant cells. So this recombination. Specialized transduction first discovered in coliform lambda. Unlike the generalized mechanism where that can package and transfer any bacterial DNA, here in specialized transduction, it is limited to transfer of specific set of genes, right? Specialized transducing particles arise when viral and bacterial host DNA are encapsulated as a hybrid molecule. So specialized transduction is carried only by temperate bacteriophages, which undergoes only lysogenic cycle in donor cell, not lytic cycle, right? So here, first temperate bacteriophage enter the donor bacteria, right? So then its genome get integrated into the host cell DNA at certain locations and remain dormant and pass from generation to generation, right? When such lysogenic cells are exposed to certain stimulus, like some chemicals or UV light, it causes induction of virus genome from host cell genome and begin lytic cycle. On induction from donor DNA, this phage genome sometimes carries a part of bacterial DNA with it. So this bacterial DNA lies on sides of integrated phage DNA and only carried during induction. When such bacteriophage carries a part of donor bacterial DNA, in fact, new bacterium, it can transfer that donor DNA fragments and form a new recombinant cells. New recipient cell means recombinant cells. Lateral transduction. This is third mechanism of transduction recently discovered. Lateral transduction. It was discovered in temperate fogs of Staphylococcus aureus. Unlike generalized and specialized transduction, in this process, it doesn't appear to be the result of erroneous phage process, but it seems to be the natural part of phage life cycle. The key here is that staphylococcal prophages do not follow a typical lytic program, but instead excise late in their life cycle, right? So this result in a mode of transduction that transfer bacterial chromosomal DNA at frequencies at least 1,000-fold greater than previously observed, right? So this is all about transduction. Thank you.